I am an unusual homeopath. I came out of the engineering world. I have a bachelor's of mechanical engineering, a bachelor's of material science and engineering, a master's of material science and engineering. And I was working predominantly in space satellites and fiber optics. In fact, was appointed to corporate working boards at Alcatel, which is where I was working when I first stumbled upon homeopathy. And I was a scientist for 14 years before I even knew of homeopathy. A colleague of mine delivers a software program, and his assessment was that most homeopaths had about a 15% success rate. Mostly, in my opinion, because they did not use what I would call scientific rigor to both assess what remedies somebody needs, as well as to determine if they had the right remedy. Most homeopaths, I think, are self-fulfilling prophecy type people. And so this has been a problem from the beginning. I look at things very, very differently. There's a small group of homeopaths, which are what I would call a scientific homeopath. It's a group that I belong to, but there's only like maybe 5 in America that I know of. This is not the first time that we've gone through this. The USPTO basically's position was that homeopathy is quackery. Their original position was that they will not issue a homeopathic patent. And then there's a website by Stephen Barrett, which basically they reference in one of their rejections. Similar to you, their statement was this ingredient is deemed to read upon water, meaning that there's nothing in it. And basically referenced a website which talked about homeopathy, the ultimate faith. So we did a presentation which is very similar to the presentation that we're doing now, although this was done in 2016. So we've gotten a little better over the last 4 years and learned a little bit more. But we went through the presentation, and in fact, similar to you, it wasn't just the examiner, but her supervisor and the supervisor of the art unit, who we needed to have a conversation with in order to get this through. And so we had a conversation with both of them. And at the end of it, quite frankly, the supervisor, McKelvey, basically said, excuse me, but it could be placebo effect, and these are extraordinary claims, and they require extraordinary proof. But they did agree to review them. So we get on the call and the next call, and I start going through. So we provided all the information to them. I get on the call for the next call, and Supervisor McKelvey basically interrupts me. He's like, I need to tell you something. And I was very much expecting him to say, this is a waste of time. There's no way I'm going to give it to us. But in fact, what he said to us was, you've convinced us, which was possibly one of the, I've been dealing with this, being a scientist in a world where no one believes what I do for a long time. And it was a very important point when I thanked him profusely, because what he had done, I said, thank you very much for looking at the data based on the data, not just based on preconceived, homeopathy's quackery. And I was very, very thrilled, and it was just a lovely day. So I'm going to start by going through some of the old pictures that I had from when I first started being a homeopath. This is a 19-year-old girl who had cystic acne for 2 years. And trust me, this woman had tried everything. And she comes in, and this is what she looked like 4 months later. And the concentration of what I gave her was a 200C, which is roughly 10 to the minus 400th, which is very below Avogadro's numbers, as everyone would imagine. Same thing here. This is one of my first patients, and I can remember the parents both came in, but the father just looked at me. He's like, I don't believe in homeopathy. And I'm like, I don't care. I mean, I didn't care. And so I gave it to him, again, an infant, and they had tried everything. The baby was 6 months old. They tried everything to try and resolve it. They're like, okay, we'll try homeopathy, because we quite frankly will try anything. This is the baby 3 months later and 5 months later. The face cleared first, and then the rest of the body. And then this one was very interesting for me, because I didn't actually think it would work. The parent, I don't know why, did not want to put a helmet on the child. This is plagiocephaly, where, as you can see here, the head is malformed. And it was a relatively old baby, and I was worried about the fontanelles, meaning the hole in your head, closing up. And I told the parent, I said, I don't know if I can do this. And if we don't see a change within a month, I will basically require you to go see someone else, because I don't think this will necessarily. And let me make a point. These are standard measurements done by physical therapists. So I basically, as you can tell, this is a 7.5-month-old. The baby should have been much stronger by that age and was not sitting up, which is a delayed, the milestones were delayed. This is the baby a month and a half later. Had reduced the plagiocephaly by 50%, down to 15 millimeters. So now I'm going to turn myself to OcuMend. OcuMend is manufactured using the same method in the claim. Namely, it's a method of producing a hydrophilic homeopathic aqueous substance, which we call HASA, gel matrix. The method comprising the steps of combining a homeopathic compound comprising arnica of a potency of at least 30 C, water, and at least one hydrophilic gelling agent to produce a HASA gel mixture, and thereafter forming the hydrophilic HASA gel matrix using at least one of a cross-linking agent or a polymerization agent. This is basically, this claim was based on us having already made OcuMend and developing the claim to cover what we had discovered. So at this point, what I would like to do is introduce Dr. Roy Geronemus. Dr. Roy Geronemus is a past president of the American Society of Dermatological Surgery, which is one of two premier dermatological societies within the United States. Past president of the American Society of Laser Medicine and Surgery, previously chief of dermatology and laser at NYU, which is, I looked it up, currently ranked the 4th best medical school in the United States. President of the New York Dermatological Society, ASDS, Segment Award winner, which is their premier award they give out once a year. Got a bachelor's from Harvard, an MD from the University of Miami, and a board certified by the American Board of Dermatology. At this point, I will turn the audio portion over to Dr. Geronemus. Thank you, by the way, Dr. Geronemus, for agreeing to be on this call. We very much appreciate it.
My pleasure. Let me start by saying that I began as a skeptic of homeopathy. I had limited experience. We had tried various different remedies over the years, and just by background, my practice is in what we call dermatologic surgery. So I do laser procedures, I do Mohs surgery for skin cancer, and I do a lot of cosmetic dermatologic procedures, including injectables, which would mean Botox or botulinum toxin injections and injectable fillers. So there are plenty of opportunities for bruising in our practice, and it's a big problem for us in terms of recovery time. And I also work in Manhattan, where patients are very demanding, and any little bruise is a travesty and requires them to hole up in their Park Avenue apartments and not be seen until they're absolutely perfect. So we've been looking historically for the answer for bruising. And as a laser specialist, I published an article around 2010, 2011 in the Archives of Dermatology, which is now known as the Journal of the American Medical Association, Journal of Dermatology, which is a major peer-reviewed journal on the laser treatment of bruising. So we came across this technique, which worked extraordinarily well. And what happened was, well, our protocol was patients would come in, have a procedure, we'd have them come back at 24 or 48 hours, and we'd perform this laser procedure, and the bruising would miraculously disappear. It was just absolutely remarkable, and oftentimes within hours. So as our practice grew, and I'm with a practice of 17 different physicians, we would notice that our schedule would fill up with patients coming back and automatically booking their appointments for 24 to 48 hours for these laser treatments, so they wouldn't have any imperfection in terms of bruising on their faces or other parts of the body. So going back a couple of years, I was approached by somebody from the Cerner company about OcuMend, And I poo-pooed it. I said, look, I've tried homeopathic regimens in the past. Nothing has worked for me. And I shooed them away. And a year later, an employee of the company whom I knew from a previous relationship at another company where we worked quite successfully together said, look, you got to rethink this. You've got to try this. It really works. So we began applying the OcuMend post-procedures, post-injection of botulinum toxin, post-injection of injectable fillers like Restylane or Juvederm, post-laser procedures, post-radiofrequency procedures, post-surgical procedures. And what was fascinating to me and the colleagues in my practice was all these patients who were filling up our schedule to come back for laser treatment of their bruising were disappearing. They didn't have to come back anymore. We were putting this dressing on, and they weren't getting the bruises they were getting, requiring the laser treatments with the procedure that we had published and developed. So I went from a total skeptic where I wouldn't let them in the door to a regular user, and I believe Dr. Polich might say that I was one of the larger users of the product in the country. We also found other unique indications. Some of you may have older relatives or know people who tend to get bruising on their arms or their legs or their hands, part of what we call senile purpura. As people get older, they have a tendency towards spontaneous bruising. So we've had patients come in who literally wanted to buy out our supply of the OcuMend because they found it helpful for that type of spontaneous bruising, for which there's really nothing else that one can do. The laser treatment is not particularly practical if one is getting recurrent bruising. So I became a significant believer. I've talked about this at national meetings. I recall, I guess 2 years ago, at a meeting up at Harvard where the issue of bruising came up, and a well-known physician, Dr. Tina Alster from Washington, who handles many of the celebrities and politicians in Washington, got up to talk about the OcuMend dressing, and I raised my hand and said, look, this is my experience as well. So there began to be this consensus amongst key opinion leaders that, in fact, this was something real and this is something integral to clinical practice, especially in areas where people are dealing with bruising. Subsequent to our initial work with bruising, I began to do more and more with post-procedural application. So we found it minimized swelling after procedures and facilitated recovery. So there was more value to this than just bruising as we thought initially. So we use it across the board in our practice for anti-bruising following injections and following many procedures that we do. The OcuMend is typically applied by our ancillary staff. Sometimes I'll do it myself, but I typically work with a team of assistants. So I have nurses and medical assistants who apply. It's very simple. It's basically putting this impregnated material in a hydrogel on the skin. It's very soothing, and it works quite well when applied by anybody. What's an interesting finding that was brought to me by this Dr. Alster, if you referred to in Washington, where many of these cosmetically minded patients don't want to walk out with a patch on their face. So what she recommended, and I didn't think this was possible, she would have the patients put the dressing on the back of their neck so it would not be visible below their hair. And yet, she would still see the benefit in her patients. So this is something that we found very helpful for over the short term for those people who didn't want to walk around with patches on their face. So the number of patients that I've used OcuMend on, certainly thousands. Feedback has been extremely positive, and we've had no adverse reactions from it. It's a totally benign problem. We've not seen allergic contact dermatitis. We've not seen allergic irritant dermatitis. We've not seen bruising get worse or inflammation get worse. It's really in many ways the perfect product in terms of the benefit-risk profile. And that's something as a physician I always look at, when I talk to patients about drugs or procedures, I talk about the scale of of justice, so to speak, where you have the benefits have to significantly weigh the risks. In this case, the benefits are high and there are no risks. As a physician in the field, I work a lot with the various different companies in the space. They often come to me, since we do have our own research division, and they look for new and exciting products or devices for acquisition. So, they're always on the look. They can't develop everything themselves. So one of the companies that came to me for my opinion was Allergan. Allergan is a multinational company, is a multi-billion-dollar company, which actually incidentally was just acquired by AbbVie a few months ago. Allergan is an Irish-based company, and they did their due diligence on OcuMend, speaking to many different physicians across the country, or the U.S. country, and perhaps beyond, to get their opinion on the product. And the response was overwhelming. So they decided to acquire the Cerna company as part of an effort to build up their portfolio. As someone on the medical advisory board of Allergan, I can tell you that they go through significant due diligence. My understanding of this process took 6 months to a year, and they worked extremely hard to confirm the safety and the efficacy and the value of this product. So I think the imprimatur of Allergan acquiring Cerna is quite significant, because they are the real deal. They're one of the largest cosmetic companies and medical companies in the world, even larger now that they're part of AbbVie. So now it's a multi-billion-dollar company. So the fact that they saw what I saw and decided to put their money where my mouth was, I think is something that really validates this whole concept. And I'm open for any questions.
One, to Dr. Geronemus' point, one of the things that Allergan did based on history is they made sure they spoke to physicians which had no connection to the company, because prior transactions they had found sometimes when people had connection to the company, there's a transaction they did which they got burnt on because everyone had invested in the company. Dr. Geronemus had not invested in the company, I should be very specific on. And basically Allergan made a point of talking to people that had no connection to the company. So other independent confirmations, Dr. Donofrio, who was again the past president, actually I think she was the president in 2017, I believe, of American Society of Dermatological Surgeons. She was also a featured speaker in AAD, as well as Dr. Geronemus, of course. Dr. Wendy Lee, who is a plastic surgeon and was the chair of both the 2015 and 2017. And again, that's a premier society for her area. We basically didn't really launch this product. We didn't bother trying to really get any customers. It was really about getting the top doctors and really getting an acquisition. We didn't have a whole lot of money. And so because we were poor, and also because of the fact that I believe people can tell the difference between somebody that's paid versus somebody that just does it because they care. And so we don't pay for any endorsements, which is unusual in this space, because these physicians often bill out at somewhere between $650 and $1,500 an hour. And the next video that I'm going to show you has top doctors in it. It has Dr. Tina Ulster, who was mentioned, who is also a Stedman Award winner and was introduced to me as the Queen of Term at one point. It has one of Allergan's top 50 account, Dr. Klein, Galderma, who is the other major company in this space, basically one of the top dermal filler trainers, top 5. And then Dr. Shudarani, who is was the person that personally treats the CEO of Allergan. So I'll show that to you now. And again, we do not pay for endorsements at all.
wanted to let everyone know today about one of my favorite products for bruising, and that is these OcuMend gel pads by Ciarna.
You know, I used to get a ton of emails from patients complaining about bruising after injections. Now I don't get any.
You can ice it all you want, but they're going to wake up in the morning with a bruise. This product is the first thing I found that works on this.
Our patients' feedback has been phenomenal. They all feel like in the morning they bounce back, get ready to take on the day, and don't look like they have the stigmata associated with injectables.
No bruising, no swelling, and no pain.
This stuff is so good that I won't do my own injections anymore without having it on.
OcuMend has been a game changer in my practice.
OcuMend is my favorite product for bruising.
These just work.
So that was obviously something we put together for marketing, but I hope you can see that these people actually are like, doctors are like, wow, this works. And again, I didn't know any of them before they evaluated it. My history is, is fiber optics and space satellites, and I didn't know anybody in the aesthetics world before I got into this. We also have a published ophthalmic plastic and reconstructive surgery. I believe you have a copy of it. I wanted to let you know that Dr. Seif was a past president of ASOPers, and then as we talked about, Dr. Lee was a meeting chair for 2015-2017. These doctors have reputations, and you cannot recommend something that doesn't work to your colleagues or you lose that reputation. In it showed 88.9% healing ahead of that expected, with 51.9% 5 to 7 days ahead of expected. We also have over 100 physician offices repeatedly buying OcuMend, despite us spending nothing, maybe $50,000 a year in marketing, in a world where it's not unusual for people, even relatively small companies, to be spending $5 to $10 million, and the big companies spending $20 million. We've sold over 100,000 pads with a money-back guarantee, and we've had fewer than 30 patients request a refund. And these guys are expensive. They typically are paying about $26 for a treatment, if not more. Candela, a medical company that does a laser treatment, that did an independent evaluation. Basically, all patients they were looking at possibly having this defined as a standard of care, and in fact but the Allergan deal happened, and so we didn't pursue that. But basically, they independently, I was not around, they had a 4-point rating scale. All patients reached 3 out of 4 improvement, with most of them reaching a 4 out of 4 improvement. And then we talked about the Allergan AbbVie. That is a top 10 pharma company. They paid $15 million for this. They're not going to spend, they may have money, but they don't spend $15 million on something that they don't think works. And they made sure they spoke to doctors who were no connection to Ciarna. These are all independent.
Mm-hmm. You see this 3-pound bar? We drop it 20 inches.
So I had to define a bruise test, which would simulate the degree of bruising that you get in plastic surgery. Because I will be honest with you, I didn't know, this is done like 10 years ago. I didn't know if it would work in plastic surgery, honestly. There was a question as to, will it work? How was the procedure? And so, not surprising, we induced the bruising on the hip. And basically, this is a placebo. We had the placebo, basically, not surprising, when you hit somebody with that bar, they bruise a lot. And this was the exact same gel, exact same treatment, 15 hours worth of treatment, 16 hours worth of treatment, excuse me. And we had completely independent blinded physicians evaluate the bruising between the two. The rating of the placebo was much higher than the OcuMend. And it's statistically significant, well below 0.000, well below 0.05. So, just to go through OcuMend, its independent data shows that OcuMend works exceptionally well. We have physician luminaries, we have research results, we have industry confirmation, most of which I had little to no involvement with. And certainly when the doctors evaluated this, I didn't really know them. I may have gotten to know Dr. Geronemus afterwards, but I didn't know him at all. And he quite frankly ignored me in the beginning. It's exceptionally dilute. It's a 50 m. It is so below Avogadro's number that it's not within any reasonable amount. And I will say, I'm a scientist first. I completely understand. In fact, I'm pretty sure that I didn't know what homeopathy was when I first used it. I used it on my toddler at the time, and something that had been the same for a year changed, which of course, just like everyone else, I tried everything in the world. I don't believe I would have used it. If somebody had told me the dilution rate, I would have said, oh my God, there's no way. But the fact is it actually does work. So with that, I'm going to now go into the science behind it, and I'll introduce Dr. Calabrese. He is a professor of toxicology at the University of Massachusetts. He's authored over 900 papers in scholarly journals, most of them about hormesis. He's author of 10 books. He's a member of the U.S. National Academy of Sciences. He's the chairman of the Biological Effects at Low Dose Exposures. He won the 2009 Marie Curie Prize, as well as a 2009 Springer Award for the International Society for Cell Communications and Signaling. He is a Peter Beckman Award winner for the Doctor of Disaster Preparedness and the world's #1 expert on hormesis. Dr. Calabrese, are you still on the phone?
I'm still here.
Okay, well, I will go through your slides and just tell me when to move forward.
Okay, thanks very much. Well, thank you very much for having me here, and it's a pleasure to share my little story on hormesis with you today. Hormesis, from my perspective anyways, it's a biphasic dose-response relationship, and it's characterized by a low-dose stimulation and a high-dose inhibition. And more specifically, at low doses, it has opposite effects than what would occur at high doses. And just from just a little bit of historical background, hormesis was first recognized, at least as a concept, not as a term, back in the 1880s, when a German physician toxicologist was attempting to discover some new antiseptic agents, and basically was using a wide variety of toxic elements, things like formaldehyde and some mercury compounds, phenol and others, supplying them to yeast in his assays, and he expected them to show a dose-dependent inhibition and mortality. And what he showed in his lab was that there was at high doses, that's exactly what occurred, but at low doses, the yeast responded much better than the control group, and they showed a type of inverted U shape of dose response. So there was like a low dose stimulation and the high dose inhibition. And he thought that he had done something wrong, but he couldn't figure out why. And so he kept repeating what they did to see if it was reproducible. They tried other aspects to see if they could still get the same results. And as it turns out, regardless of what agent they used, and they studied about 12 to 14 agents, they kept getting the same results. And so what happened here was that, now this is back in the 1880s, and there was a profound conflict at that time between what I call today's world, traditional medicine and homeopathy. And the gentleman's name was Hugo Schultz, and he was in a traditional medical school at the time, and he used this, he presented these results at a local conference, and about a year later, as a result of looking at other types of relevant information in the clinical world and experimental world, he came to the conclusion that he had discovered the explanatory principle of homeopathy. And really what happened here was that at the same time there was an extract from a plant called veratrin, And Veratrin had been used by some homeopathic physicians to treat gastroenteritis, and it was very effective. And so they published their paper. And so what happened in the early 1880s is that the causative agent of the gastroenteritis had been recently discovered and isolated, and they knew the bacteria, and the person who had isolated it was the famous Robert Koch from the Koch laboratory, not too far from where this researcher was. So he went to visit Koch, he got the bacteria, and he wanted to see if this Veratrin, this new homeopathic drug, could actually treat and kill the causative agent. And so Dr. Hugo Schultz, back in his lab, applied the homeopathic drug, and no matter what dose he gave, it did not kill this, did not kill the causative agent of the gastroenteritis. And so they had the homeopathic data, it seemed to work, but when he went in the lab, it actually didn't kill the causative agent. So he was faced with a conundrum. What should he do? Did the homeopathic drug work? Didn't it work? How did it relate to anything? So Schultz kept developed he really didn't know the answer, but in light of conversations with colleagues, he made a creative leap. And what he said was, I think that homeopathic drugs work not by killing the causative agent, but by enhancing the adaptive capacity of the individual to resist the infection. And he used his low dose yeast data to as support of that by saying at low doses you can see that there's an upregulation of an adaptive process, and high doses you see the opposite. And within this framework, he created for the first time the concept of hormesis, in which he believed that most drugs will act differently at low doses than at high doses, and can act by upregulating adaptive responses. Well, as you might imagine, given the terrible conflict that exists historically between traditional medicine and the concept of and homeopathy, that Schultz became quickly, you might say, basically persona non grata, not appreciated, ultimately very marginalized within his community, and ultimately had just a very difficult time for, he was a young professor at the time, for the next 50 years, but he persisted in this. And what happened is that the concept of hormesis got off on the wrong foot, you might say, with the medical community right off the bat, because
Dr. Calabrese, so to that point, can I ask you a question?
Absolutely.
When you started into this, were you a homeopath to begin with? What was your initial feeling about homeopathy?
Well, my initial feeling was highly skeptical, and probably beyond more negative than that. Actually, I was a total non-believer, and I would have to say I saw it as a problem for me, because I was very interested in the hormesis concept and its association with homeopathy was causing me all kinds of problems. And so I was trying to find biological distinctions and got into this in a fairly deep way. And also ultimately, what happened for me was I went down the road to try to understand hormesis. And in the course of this, I have taken hormesis a long way. Basically, because of this initial linkage to homeopathy, it became hormesis became kind of a dead in the water scientific concept, totally abandoned by the scientific community, the medical community especially. And in fact, when I really started to take a strong look at this, late 1980s, early 1990s, it had no standing in the scientific community. In the gigantic database Web of Science, it was cited essentially about 10 times per year throughout the 1980s. So that's pretty minimal, it means it just barely has a heartbeat, so to speak.
Mhm.
And so and the only way somebody like me could begin to even look at this and take it seriously was that at that point, you know, I had been well established in the field. I had had many books published, many hundreds of articles published, and so I had a tenured full professorship at my school, so I could actually take a risk with something. And so I wanted to pursue this hormesis thing because I had seen it in the laboratory myself, and I knew that there was a good scientific foundation for it because I had spent a lot of time personally on it. And so, as it turns out, as a result of I think really my leadership on this, the scientific community over the last 30 years has taken closer looks at the hormesis idea. And initially, what we had was we had a lot of observations that were reproducible in a wide range of biological models, from bacteria all the way up to humans, and really everything in between. It didn't make any difference what endpoints you were trying to measure. It didn't make any difference essentially the cell type that you were studying. It seemed to occur, but I kept being always criticized because in the world of toxicology and pharmacology today, what I'm told is that we worship at the altar of mechanism. And unless you can explain things mechanistically, you can't get published in the leading journals. You just can't do it. And so if I was to make that step, I had to do that. And I'd have to say that I've really tried to answer all the questions and all the challenges that the field has placed on me. And in 2013, I actually published a paper in one of the leading toxicology journals with 400 different hormetic dose responses with the articulation of the mechanism down to the receptor and the cell signaling pathway. And now it's out to really over 1,000, more than that, mechanisms for specific hormetic dose responses, very comparable to what you would find for any type of pharmaceutical agent from all the major pharmacological journals. And so when you take a look at now the field of hormesis, you find that we went from having 10 citations per year to this past year approaching 13,000 from all over the world, all different disciplines, all seeing the same thing. And now in the last 8 or 9 years, it's made it into as a central feature in major toxicology and pharmacological textbooks. So it's really made it now into the literature, and major federal agencies in the U.S. are funding hormetic research. I've been funded by the U.S. Government for the last 25 years at substantial levels of funding. So this has come a long way, and what's actually happened, and as it's emerged, many things have developed, and I'd have to say has actually drawn me back to taking a look at its relationship to homeopathy, something that I had rejected earlier, because I look at the dose ranges over which this low dose stimulation, low dose adaptive responses occur, and many of these studies are well, they're in whole animal studies, but a lot of them are in cell studies as well. And what we find is that well, I generally look at hormetic responses within the 10 to the minus 9 to 10 to the minus 14, 15. But I have a number of examples where hormetic effects with the good mechanistic understanding have gone down to 10 to the minus 18th to out to 10 to the minus 21 molar. And in the 10 to the minus 18th study, which I've really looked at in considerable detail, this particular agent was able to induce very substantial hormetic effects with only one molecule per 3,300 cells in an amazingly highly dilute situation, and with actually some interesting follow-up mechanistic work trying to understand, how this was mediated through certain receptors and other aspects, and so essentially, and I've built other supportive studies along those same lines that essentially lead me to have begun to try to test whether we can push this biological activity down to and past the Avogadro number. So based upon what I'm saying here is that I really see no indication that this low dose stimulation could not occur at or below Avogadro's number. I think you just need to have more people beginning to look at this, take a look at various cell types and endpoints, and really do a deep dive into some of these unique but very sensitive mechanisms that we're seeing now occur. So I'm seeing a scientific convergence of hormesis and some elements of homeopathy, and I don't know where it's all going to lead, but it's leading to the study of much lower doses and concentrations than were ever believed possible, and approaching and perhaps even exceeding Avogadro's number. And there's no reason for me to think that it's not possible.
You know, I'm an engineer and I really care about data. You know, we've got the data that it's effective in people, and when, you know, and it's all about matter of funding and having the money actually to do the research, which at this point there isn't. But that is absolutely one of the great plans that we have with our product, is to use that money to do the research, because right now nobody can get the funding to do Avogadro's number. So I'm now going to switch to talking about what we saw, and perhaps, again, not being bound by theory, what we think might be happening down at the 10 to the minus $100,000 level. I mean, not dollar, concentration. So traditional homeopathic remedies are often dispensed in the mouth, and they're also placed in water, stirred, and drank. Again, potencies, you know, highly diluted substances, 10 to the minus 60th or whatever. And so I started into this not ever wanting to start a company. I was a practitioner. I knew it worked better topically, and I was looking for a gel. And what happened was, when I was practicing, I was perplexed by why there was only all the homeopathy, you know, we were orally dosing high potency, meaning highly dilute substances beyond 30C, more dilute than 30C, but we couldn't find any in a gel, and I didn't understand why. And so back when I was a practitioner, I made one and it didn't work. And by my background's particularly material science, and I have no idea about anything on drug development or anything. And so I just didn't understand that. I'm like, why should that not work? And so I learned through time that traditional formula is to add it to a gel base. And because of homeopathy being something that's very tradition, basically original salves were designed back in the 1800s, and they just added it at the end. And actually, it's a very common way drugs are. And just talking to other people, they were, you know, because we don't really understand it very well, they were worried about, you know, if you add it earlier, what happens if something in the process of making it causes problems? So pretty much everything was done where you added the gel at the end. And as I mentioned, we tried to make it and it didn't work, and I didn't understand why. After working with it, again, I've got a background in material science, I came up with a theory that, and this is after observing data, that we could make it if we added it at the beginning and then cross-linked. And if we added it at the end, it didn't work. And so I basically came up with a theory. I said, there must be a structure which is made where the water, what I did know is, again, two of my degrees are in material science. What I knew was that the reason that water, so if you think about it, it's like, why does the water not fall out of the gel, right? And the reason is that the gel holds the water through hydrogen bonds and keeps it in place, which is called binding. In the world of gel, they call it bound. The water's bound. And so I had a theory that there must be a structure there that is formed when you have the water. And when I'll call it normal water, I mean, there's a lot of different things. Sometimes Jerry Polich calls it, bulk water, but it's really water that's able to be moved. So I'll just use the word is normal water for lack of a normal thing as I go through. But basically, I presumed that you needed to have normal water that could move, and that when it was bound, it wouldn't form any nanostructures. So this was just something, it's funny because during the Allergan deal, I had to go back through my notebooks and share it with them and my research notebooks. And so months later, I actually learned of Jerry Polich's work. And one of the things that's interesting is water is really unique. You can see it forming this bridge. There's no other substance that'll do this. And so the thing is that nobody really understands why. And I'm a materials engineer, and I've seen a million phase diagrams. A phase diagram for water is the most bizarre looking thing I've ever seen in my entire life. And so Jerry Polich came up, and others came up with an idea that there must be ordered semicrystalline structures formed around hydrophilic substances and particles in water. And so what's interesting about it, what you see here is that it's a structure, it displays solutes. So you see this region here, hopefully you can see what I'm showing, is what they show is over time, this is a time, it pushes the solutes as it forms out. So this is a line where here's the solutes. Can everybody see my writing? And here's without the solutes. The same thing happens as you get a gradient on pH. And this has been verified. The solute displacement has been verified by 11 independent groups. It's confirmed by X-ray scattering and collaborated by a simulation which used van der Waals forces to simulate what they think is there's two different structures, one that's tetrahedral and one that's close packed. So that gave me, okay, there might be something going on with the water. So that's one piece of it. And then the other thing that I got years later, my invention was in 2011, early 2011. And it was published in 2012, but I didn't know about it until 2015. And what these two different researchers found is looking at atomic force microscopes, which, let me remind you, are something that was not around, I think, 30 years ago. So it's something that was not available back in the day. And what they show is these structures, and that they saw to dilutions of 10 to the minus 18th, okay? So we have visual things of these water structures, and they seem to be completely of water. Then Lowe shows that, so what you see is with the solid line here, see basically what you have is pure water or normal water. And then strangely with the nanostructures where you see these peaks that are not there with normal structures. Okay, so there's scientific indication of the fact that these things, these water structures, have an effect. Konolov basically did dynamic light scattering where he showed a substantial amount of these structures down at concentrations of 10 to the minus 19th, which, let me remind you, have one molecule of original substance and to 10 to the 19th molecules of water. And you really shouldn't, this is so far below detectable limits that you really shouldn't be seeing any peaks on dynamic light scattering at this level. Konolov further showed that it had an effect that these, again, very dilute substances had effect on long-range things such as surface tension. What you see here is, as we would expect, in the absence of the nanostructures, you see the water, you see the surface tension plateauing, as you would expect. What you don't expect with the presence of nanostructures is you see these peak changes, which are completely unexpected. Same thing with conductivity. You plateau once you get to lower concentrations. However, in the presence of nanostructures, you see changes, which means you have to have, well, this is not my area of expertise, but it suggests that there's enough of them in there that they're having effect on relatively long range, large things. So, in summary, we have a significant number of people who overwhelmingly, independently verify the efficacy of an exceptionally low concentration product. And the bottom line is, it either works or it doesn't work. We don't know exactly why. Research hasn't been done because every researcher in the history of the world that has ever tried to go below Avogadro's number basically gets no funding and is laughed out of the world. However, hormetic reactions at low concentrations suggest something's going on there. Moreover, as the world's expert, which I am grateful that Dr. Calabrese agreed to be on this call, suggests that, you know, there's probably not an edge at Avogadro's number, and we need to investigate it, but people really haven't because of the experience of Schultz that he talked about. There is data that shows that the HASA nanostructures change the properties of water. And without being bound by theory, what I will tell you is the two things I know are, one, that it works, and two, that it works when you add the active ingredient before you cross-link or polymerize, and it doesn't work if you do it afterwards. I believe it's nanostructures. It makes sense to me, but until we do the research, we don't know 100%. But that is a reasonable theory as to what's going on there. And it was really no one understood really why homeopathy worked. And therefore, they really didn't understand that there was a need to do this. You know, it's like, oh, adding at the end should work. I actually thought it should work at the beginning, and learned through experimentation that it didn't.